Thank you all so much for coming. Kind of a last minute crop tour. This all started with a picture that I posted on Twitter that looked really good and got some people excited about coming to see the plots. So that's just wonderful to have you all out here. This trial is hosted by Starvation Farms, so big thank you to them. And yeah. And so these trials are part of the Resilient Dryland Farming Alliance. So this is a stream of federal money that comes to our station. It's a collaboration with the USDA ARS. And with that, I want to introduce my counterpart on this project, Dr. Kate Reardon, microbiologist.
Hi, I'm Kate Reardon. I'm a microbiologist with the USDA in Adams, Oregon, and I'm collaborating with Christina and our OSU colleagues on the Resilient Dryland Farming Alliance. And where I start to come in this project is following up after the cover crops, after the alternative crops, and looking at what happens to the rhizosphere and the wheat. Here you can see a sample of all this root impacted soil. And taking it apart, the soil is just really tightly associated with these roots. Well, to tell you, this is where the party is. This is the action, and this is where I'm going to be looking at how these different crop rotations impact the microbial communities and what kind of influence they're having on the nutrient cycling, and also, you know, are we seeing any difference in our beneficial communities?
The goal here is to understand cover cropping and alternative cropping in dryland systems. And so we'll start with the cover crops. So this first square that's marked by the bicycle flags is our cover crop trial. We've got fall seeded treatments and spring seeded treatments. And so in terms of cover cropping, we all know that there are trade-off benefits and trade-offs, and understanding how that might work on your operation is a pretty complex decision-making strategy. So some of the things that we're interested in the cover crops in particular is water use. So in the center of each of these plots, we've got fiberglass tubes that weekly we send a sensor down, and we're able to get soil moisture profile data as the season moves along. And so one of likely the most valuable things that we'll generate is understanding the right time to terminate your cover crop to preserve moisture in the soil profile for the cash crop. So a couple things about this trial. So we planted this with, we're in no-till conditions. We used a Fabro drill with 12-inch row spacing. And for the cover crop, we tried to manage it like a producer would. So very minimal inputs. We did seed bed preparation with glyphosate, basically chased the drill with a glyphosate application, and we don't even have seed care in the cover crop trial. And so without seed care, I'm a plant pathologist, so I can't help myself. I'll pass around some loose smut of barley. There is another one around here. So this is typically taken care of with our seed care packages. Breeders really don't breed for smut and bunt resistance anymore. We rely heavily on seed care packages. It's a seed-borne fungus. And then these spores on the black spores on your fingers, kind of interesting. So I'll just go ahead and pass that around. So Okay, so we are crop year 10.5 inches of annual precipitation since September. Pretty impressive, especially compared to the very low rainfall year that we had last year. So we'll just kind of walk through these treatments, and then after I wrap up, you're welcome to just kind of poke around in the plots because they're just showing really nice right here with the rainfall that we've had. So we've got a fall mix, lightning winter barley, which was bred by Pat Hayes, Austrian winter peas, and then brassica. So the peas and the barley are really tending to predominate in this mix. A lot of different theories on cover crop seed mixtures. Kind of a nice way to hedge your bets in terms of seed size, seed depth in your operation. To have the a species predominate, you can really learn a lot by planting a mix. Let's see, fall planting was October 28th, and then our spring planting was March 14th. And so when we're thinking about terminating these cover crops, we need to have that balance of getting the right biomass, not depleting soil moisture, making sure the crop is in the ground for long enough to have your NRCS check mark. And okay, so this is yellow mustard, Ida Gold. Brassicas, as we know, can have some biofumigative properties in the soil. So essentially what we see is reduced soil-borne disease of winter wheat following brassicas. So that's kind of the interest in planting a mustard. We'll move on down. Phacelia. So Phacelia, the cultivar the variety name is Super Bee. So this is kind of a pollinator friendly species. There is interest in planting pollinator buffers. So potential to kind of have a twofer there with a cover crop that does count for a pollinator profile. And we haven't had, I encourage you to come in and really take a look here. So this is the plant itself. Not the best stand, but you can pass that around. A lot of what we're doing is trying to understand what works in these systems and what doesn't work. So we take data from the year and evaluate, and maybe we have some winners here and maybe we have some losers, but we're trying all kinds of things. Common vet, a little better stand here. Spring planted. Pass one of these around. And then all of this is, of course, in comparison to fallow. So our next treatment is just a chemical fallow. These plots will be planted to winter wheat in the fall. We'll take off the winter wheat crop and understand what happened to yield, protein test weight, disease in comparison to fallow. So spring planted tillage radish. We got some folks here that have had a lot of experience planting tillage radish. The idea is you get this root mass that can really help break up compaction. We won't get enough biomass in a spring planted situation to have that effect at this rainfall location, but we're doing this exact same study at CBARC in Pendleton in a higher rainfall zone. Some of the treatments have higher biomass in Pendleton. Some of them have higher biomass here. So kind of understanding seeding timing, soil temperature has a lot to do with it, with what we're seeing here.
Christine, are you
Yes.
terminating all these this week?
So Chris is asking about cover crop termination, and we're gonna terminate just the winter seeded treatments this week. Would I like to keep them in the ground longer? Yes, but basically what we're fighting is that panicle development of downy brome. So we know that at about 1,000 growing degree day units starting in January, we've got viable downy brome seed. So as we approach 1,000 growing degree day units, we got to be careful that we hit our herbicide timing prior to viable seeds. But they could go longer if we had higher tolerance for the downy brome. Spring mix. So this is spring mix, and again, that planting date was March 14th. Austrian peas, spring mustard, a spring barley, the Phacelia, that pollinator species, tillage radish, and common vetch. I think it's a little bit early to tell what exactly is going to predominate in this species mix, but we come in and we sample prior to crop termination to understand, the ratio of species. We know the ratio that we planted, but we want to know the ratio that we end up with. So this is winter pea, seeded October 28th. This is a numbered variety by Dr. Rebecca McGee, pea breeder for the USDA ARS. Huge amount of biomass here. You can see the volunteer wheat is poking up through that canopy, but we're getting row coverage even on our 12-inch row spacing, and huge potential for weed competition. And then the really interesting thing to me that I want to point out here is our nodulation. So I'll pass this around. So you can see these nodules that are pretty impressive. Don, you want to pass that around? And so I had to write down the protein, but so these root nodules are pinkish. And so they've got a protein called leg hemoglobin. You probably all know this, but it's a protein that carries oxygen, just like what makes your blood red, carrying oxygen. So that pink hue carries oxygen so that, Curtis, help me out, you can fix more N. So a lot of what we're trying to understand here with our nitrogen fixing cover crops in particular is what kind of nitrogen budget they might leave over for our cash crop. And so what we're doing is taking tissue samples, soil samples, and working with UC Davis and their lab to get a nitrogen budget so a grower can understand if they could even pull back in the fall on their urea application for the cash crop. The peas are seeded at a goal of 20 seeds per square foot, which is huge. And our lentils are at, I gotta look that one up. High seeding rates. We learned a lot in our first year, so we've got 2 years of good data under our belt at this point. We'll go a third year and then kind of get together and figure out what our main takeaways and kind of design some differences moving forward. So we'll plant this to winter wheat in the fall, and then take data and understand, yield differences in our fallow controls compared to cover crops.
Awesome.
Things like that. Yeah, I'm excited for it. So this is going to be terminated Thursday or Friday if we get a spray window, but if not, next week. Spring barley, barley is known as a really great weed competitor and can put on biomass and really, tiller out pretty quickly. This spring barley is still looking a little beat up, a little yellow, and I think that, you know, we had 34 pounds of available nitrogen in the soil. We did not add any fertility at the time, so this has not received any fertility. My best guess is we're looking a little beat up because of the cold and you know, hitting those nitrogen resources. Winter lentil, variety Morton. The lentils look quite a bit better than they did last year. And, you know, as you'll notice, especially with the, you know, kind of slimmer plant architecture without that competitive ability, you'll notice that we've got a lot more volunteer wheat within this cover crop treatment. You know, volunteer wheat, is that a bad thing in your cover crop? Not necessarily, but it's just kind of one more consideration, especially when we're thinking about termination date. And then just because it's on the top of my mind, so you'll notice we have cultivar names for most of these treatments. As cover cropping you know, becomes more popular in the PNW, we're seeing seed come out of the Midwest that is variety not specified, or VNS, which is not necessarily a bad thing if you're working with a reputable seed source. But it's something to really be mindful of if your seed is coming out of the Midwest. Another thing to mention is storage conditions for seed in the Midwest are very different than in the PNW. So we need germ tests that are within 6 months, no matter where your cover crop seed source is coming from.